In this video, we will discuss about DNA. So, what is DNA and how DNA is formed and what is the structure of the DNA? In this video, we will discuss about that. Do you know there was a time that DNA is a very tough topic to study because doctors don't know much about that. But now we almost discover the DNA. We know much about DNA, and DNA is the first and foremost topic that we should study. And now, what is DNA first? DNA stands for, we know the name first. DNA stands for DOC, ribose nucleic acid. DNA stands for deoxyribose nucleic acid. This is just the name. Okay, and let's suppose this is a ovum, or you can say egg, from the woman egg, and we have one more thing, and that is sperm. This is the sperm from the man, or you can say sperm from husband, and ovum come from the wife. When the ovum here fuse with the sperm or meet the sperm, and what we call, and they fuse to each other, and then this ovum become fertilized ovum. What we have right now, fertilized ovum. Now in this fertilized ovum, we have chromosome. Inside we have chromosome. What we have? Chromosome. Okay. And this fertilized ovum have how many chromosomes? 46 chromosomes. 23 come from mother. We have 23 chromosomes come from mother. And 23 other chromosomes come from father. So totally we have 46 chromosomes in the fertilized ovum. And this we call genetic material. And this genetic material we have DNA. Right? From chromosome or within chromosome we have DNA. Okay, and from DNA will express itself as RNA, or we say DNA will make copy, and then we have RNA. So RNA we will study in the next video. But now you just know DNA will make copy, and it will make RNA or messenger RNA. And from messenger RNA, we will make protein. Protein, we have different types of protein. Some protein will make the structure of the body, and some protein will make the function of the body. Okay, so it means first we need to understand DNA, or we learn about DNA first. Now, what is the definition of the DNA? I will give you the simple definition of DNA, or the very basic definition of DNA. And DNA is a linear, now you must know, DNA is a linear polymers of what? Of nucleotides. DNA is a linear polymers of nucleotides. So you need to remember this word, nucleotides. Okay, so this is the simple definition of the DNA. Then how to make DNA? So to make DNA, we need raw material, or we can say we need ingredients to make DNA. Or what is the component of the DNA? Now, we will talk about the raw material. We will talk about raw material to make DNA. So first, to make DNA, we need nitrogenous bases. What we need? We need nitrogenous bases. And what is nitrogenous bases? Nitrogenous bases, you can say nitrogen with carbon. What? Nitrogen with carbons. Okay, nitrogen with carbon, then we say nitrogenous bases. And nitrogenous bases, usually we have two types. And what is that two types? So number one we have double ring, double, and we have single ring also, single ring. If you have two ring like this, one, two rings like this, we call double rings. And if we have one ring like this, we call single ring. Now, double rings, we have bill rings. I will write here. Double ring, we have bill rings. And single ring, we have by remeaning. Okay. And bill rings, we also divide this into two types. And number one, we have, you need to remember this name, adenine. We have adenine, and we have guanine. And pyrimidines, we also have two types. And number one, we have cytosine. What we have? Cytosine. And number two, we have thymine. Okay, so this is the nitrogenous basis, or we can say the raw material to make DNA. You have adenine, guanine, cytosine, and thymine, but not enough. What we need more? I will remove this. What we need more? Now, we need other things to make DNA also. Or we need another raw material. And then we need sugar. What we need? We need sugar. And what is that sugar? So that sugar we call benthose sugar. What is benthose? Benthose sugar. Actually, benthose is enough. Benthose means sugar already. But I put sugar here, then you can understand easily. Now, what is benthose sugar? Benthose sugar is the sugar have 5 carbons. How many carbons? 5 carbons. So let I make the structure of the benthose sugars. The structure will be like this. This is the structure of the benthose sugar. And we have 5 carbons. Where is that 5 carbons? Carbon number 1 will sit here. And this at this corner. And carbon number 2 will sit at this corner. Carbon number 3 will sit at this corner. And carbon number 5, number 4 will sit at this corner. And carbon number 5. Carbon number 5 is not sit here. Carbon number 5 will sit here. We call C5. And here, what it is? Here is a oxygen. And benthose sugar also have two things more. We have what? Hydrosol. So we have double hydrosol. One hydrosol, what we call hydrosol. So one rosol sits at the carbon number 3 and one rosol sits at carbon number 2. So we have double hydrosol. So the sugar with double hydrosol, and the name of this sugar we call ribose sugar. What we call? Ribose sugar. Actually, ribose sugar, this sugar we will make RNA. What we will make? RNA. But we will learn or we will discuss about RNA later. Now we will focus on DNA. And how to make DNA, then what we need? We don't need ribose sugar. Okay, we need another sugar. And what is that? So now if you remove one oxygen here, this is hydrogen and oxygen. If you remove one oxygen and this sugar will become, the structure will become like this. Now we have C1, C2, C3, C4, okay, and C5 here. And now we have As carbon number 3, we have one hydroxyl. And here, we already removed that oxygen. And then, now we have at the carbon number 2, we have only hydrogen. So this sugar, the structure of this sugar we call deoxyribose sugar. Deoxyribose sugar. So this sugar we will make DNA. You see, D means remove. If you study Latin, then you know D means remove. Okay, and oxy here means oxygen. Okay, and ribose sugar, the sugar have 5 carbons. Okay, so this is the raw material. Then now how to make it? I will remove this. How to make DNA? How to make nucleotides? Because DNA is a polymers of nucleotides. How to make that? So first, here is a sugar. I will draw again here, sugar. Then now, what we have? Nitrogenous basis. And nitrogenous basis, what we have? Adenine, guanine. Again, I will draw here. Single ring we have cytosine, and what we have? Thymine. Remember that. Thymine. And now when we bring this adenine, then we fill with this sugar, and then this. Now we bring adenine. Adenine, then we fill with the sugar. Okay, and then here I will write sugar here. Sugar. And then this adenine with sugar, it will become adenosine. What will become? Adenosine. Continue. Now we have here. Here we have sugar also. Sugar here. And now we will bring guanine. We fuse with the sugar. We put here guanine. And this guanine with sugar, it will become guanosine. It become guanosine. Adenine or guanine with sugar become guanosine. And now continue to make it. Continue. Now we will bring cytosine and we add with sugar. Cytosine here. And then sugar. And then here, now we have cytosine here. Cytosine. And cytosine with sugar, it become what? It become cytosine. Now it's become cytosine. Yeah. And one more here. This is sugar. And then we will bring what? Thymine we put here. Thymine. Now thymine with sugar, and now it will become thymidine. Okay, now we have adenosine, guanosine, cytosine, and thymidine. Then now adenosine. So this adenosine, what we call, means sugar with adenine. This what we call nucleoside. Okay. So these things we call nucleoside, not nucleotide. Nucleoside. Nucleoside means sugar with adenine, or sugar with guanine, or sugar with cytosine, or sugar with thymine. Now to make DNA to become nucleotide. So what we need now? We need nucleotide. But now we have nucleoside. Now what we need? We need nucleotide. because nucleotides is DNA. Nucleoside is not DNA. DNA is nucleotide. So now, how to make nucleotides from nucleoside? Very simple. We will bring one phosphate molecule. We will bring one what? Phosphate molecule. Phosphate molecule. So what is phosphate molecule? The phosphate molecule is a molecule of energy. Means now we will bring, I will make here. This is sugar. This sugar. And what it is? This is adenine. I just take only example of adenine. Adenine here. Now you know already that adenine with sugar become adenosine. Now sugar here. Sugar here. Now you will bring one phosphate we will put at the phosphate energy. Okay, phosphate is the energy molecule. Now you know here we already have carbon number 1, carbon number 2, carbon number 3, carbon number 4 here. But carbon number 5 is here, not here. Now we will bring one phosphate we put here. This. Okay, and this become, if we put one phosphate here, then this become nucleotides. Okay, from the nucleosides we put one, we add one the energy molecule, and then it's become that the name of that is phosphate, and then we put here at the carbon number 5, and then it's become nucleotides. Okay, so nucleotide is a DNA. Okay, now, but we need a strand. We need a chain. We need a chain of DNA because now this is only one molecule. Now I will make it here. When adenosine add with phosphate here, then what we call the name of this, what we call adenosine. One phosphate we call monophosphate. Mono means one. In Latin language, mono means one. Okay, but if we have two phosphates here, then we call diphosphate. Adenosine diphosphate. Okay. But if we add one more molecule here, and it's become triphosphate. Tri means the 3. Triphosphate. Tri means 3. Okay. Di means 2 phosphates. So adenosine with 3 phosphates we call adenosine triphosphate, or we say ATP. ATP stand for adenosine triphosphate. This is the molecule with rich energy, very rich energy, because it has 3 phosphates here. Okay, now what we have here, these molecules need to link to each other, or we will make the bonds between the nucleotides. Here we have how many? Okay, here is adenosine. Here guanine. Here cytosine, and here thymine here. Now here is a carbon. Here is a carbon number 5 here, and we have one phosphate here. Now we have 3 phosphates here. There, the same here. I will make one more here. then you will understand. Adenine. No, here. Adenine. Warning. Cytosine. And then thymine. Okay. This we have three. Here we have three molecules of phosphate here. 1, 2, 3. Here we also have 1, 2, 3. Cytosine we also have 1, 2, 3. And here we have 1, 2, 3. Okay, here on we have 3 phosphates here. And to make a strand of DNA, strand, or you can say strand or string, strand or you can say string, or you can say chain. Okay, to make the chain of DNA, we need to bring here, and we make the bonds between two the nucleotides. Okay, between nucleotides we need to make a bonds. What we need to make a bond. Okay, bond is like an agreement. Now, here we have at this position we have C5, but here at this corner we have C3, carbon number 3. Now I will ring the adenine or adenosine here. Adenosine, I will put it here. Okay, then we need one enzyme, and this enzyme will take or cut two phosphate molecule here to make we use two phosphates as the energy to make the bonds between the nucleotides. I will bring adenine and I will put it here. Okay, so at the position or the area or the corner of the carbon number 3. And now between that we have one bond. Okay, how we have this bond? We take the enzyme will take these two molecule of energy phosphate, and then we will make the bond here. And now I will bring adenine. I put here. This is adenine. And then next I will bring warning. I will put here, and we also have a bond here. We also have a bond. How to have this bond? We also take two molecule of phosphate or we say that energy molecule. We use two phosphates and to make the bond here. And then now we have what we have. Warning here. And next, the same for that, the same position, the same process. And then there is an enzyme will come and cut two phosphate molecule, and then we put here to make the bond here. And here, what we have, cytosine here. The same that process, and we have one more bond here. And what is that timing we put here? Timing we put here. Now, here we got C3. Here's also C3, and we have between the nucleotides we have bonds. And what is the name of this bond? The name of this bond is what we call the name of this bond we call phosphodiester bond. Phosphodiester bond, because we use the phosphate molecule to make the bonds. Okay, so now what we have now, so this we call the strand of DNA, or we say single. Now we have already have single strand of DNA. Single strand of DNA, because we already connect or we link on the nucleotide together. Okay, this is one nucleotide, one nucleotide, one nucleotide, one another nucleotide, and then when we make a bond or we link it together, and it's become the strand or string of DNA. But this is a single, okay, single strand. So one thing you need to remember that this for the ester bond, this bond is very strong bond. Why? Because if you change the pH environment, or if you change the temperature, or with the temperature, the changes of the temperatures of the environment, so we cannot break this bond. This bond cannot be easy broken by the temperatures, or I want to say the environment temperature. Or if we change the pH, okay, slightly, a little, then this bond also strong, cannot be broken. Clear? So there are some enzyme, the nasty enzyme, it can cut this bond. But that I will talk about that enzyme later. Okay. Now this is now we already have the single strand of DNA. Then, but we need one more strand, because single strand is doing nothing. It's nothing. Okay. We need one more strand. Then I will talk about that, how to make another strand in the next video. Because what we need like this. We need double strand. We need this. What we need double strand. So this is the structure of DNA. Okay, we need double strand, not single strand. But here we already have single strand. In the next video, I will talk about how to make the second strand, and the DNA will be completed. Thank you so much.